Hello, Bachcho. Welcome to Hello Biology. This is the fifth lecture of the chapter, Principles of Inheritance and Variations. In this lecture, we will discuss another law of Mendel. So let's get ready. Let us now discuss Mendel's second law, known as the law of segregation. This law is also called the law of purity of gametes because it explains what happens to alleles during gamete formation. To understand this law, let us recall the F1 generation of Mendel's monohybrid cross. When a pure round-seeded plant was crossed with a pure wrinkled-seeded plant, all the F1 plants were round. However, these F1 plants carried both alleles, one for round seed shape and one for wrinkled seed shape. Now the question is, what happens to these two alleles when gametes are formed? Mendel proposed that the two alleles of a character do not remain together forever. During gamete formation, the two alleles separate or segregate from each other. As a result, each gamete receives only one allele. For example, an F1 plant carries both the round and wrinkled alleles. When gametes are formed, some gametes receive the round allele, while others receive the wrinkled allele. In other words, every gamete is pure because it carries only one allele for a character. Later, during fertilization, these gametes unite randomly and restore the pair of alleles in the offspring. This is why the hidden wrinkle trait, which disappeared in the F1 generation, reappears in the F2 generation. From this observation, Mendel proposed the law of segregation. The law states that the two alleles of a character separate from each other during gamete formation. Each gamete receives only one allele. The alleles remain unchanged and reunite randomly during fertilization. So, students, the most important point to remember is that alleles never mix or lose their identity. They simply separate during gamete formation and come together again in the next generation. That is the essence of Mendel's law of segregation. This law has been called Mendel's first law by Correns. What are you saying, ma'am? At this point, you might have a question in your mind. If Mendel proposed both the law of dominance and the law of segregation, then why did Correns call the law of segregation Mendel's first law and not the law of dominance? The answer lies in the importance and universality of the law of segregation. The law of segregation applies to all Mendelian traits, whether dominance is present or not. For example, even in cases of incomplete dominance, codominance, or multiple alleles, the alleles still separate during gamete formation in exactly the same way. This means segregation is a universal principle of inheritance. On the other hand, dominance is not universal. Many traits in plants, animals, and humans do not show a clear dominant recessive relationship. Therefore, dominance is only one possible relationship between alleles, not a rule that applies everywhere. Another reason is that segregation represents the fundamental mechanism of heredity. The entire process of inheritance depends on the separation of alleles during gamete formation. Without segregation, predictable genetic ratios and inheritance patterns would not exist. Because of this, Correns and the other rediscoverers of Mendel's work recognize that segregation is the core principle underlying all Mendelian inheritance. That is why the law of segregation was given the status of Mendel's first law, while the law of dominance was considered an important consequence observed in certain situations. So students, remember, dominance explains which trait appears. Segregation explains how alleles are transmitted from one generation to the next. And transmission of alleles is the foundation of heredity itself, making the law of segregation the most fundamental of Mendel's laws. Let us now understand the law of segregation with the help of the two important points shown on this slide. The first point states that the two alleles controlling a character stay together in an individual. This means that every organism carries a pair of alleles for each character, one inherited from the mother and the other from the father. For example, in the F1 generation of Mendel's monohybrid cross, each plant carried both alleles for plant height, one for tallness and one for dwarfness. Now let us move to the second and most important point. Mendel observed that during the formation of gametes by meiosis, these paired alleles do not remain together. Instead, they separate or segregate from each other. As a result, each gamete receives only one allele of a character. For example, if a plant has two alleles for height, the gametes formed by that plant will carry either the allele for tallness or the allele for dwarfness, but never both together. This separation happens randomly and naturally during meiosis. Later, during fertilization, the gametes fuse and restore the pair of alleles in the offspring. Therefore, the offspring again receives two alleles for every character. This is why Mendel concluded that alleles do not mix with each other and do not lose their identity. They simply separate during gamete formation and reunite during fertilization. This principle is known as the law of segregation, or the law of purity of gametes. So students, remember one key line. The two alleles of a character remain together in an individual, but separate during gamete formation. So each gamete receives only one allele. That is the essence of Mendel's law of segregation. That's all for today's lecture, students. Today, we explored Mendel's laws of inheritance and understood how Mendel explained the transmission of traits from one generation to the next. We discussed the law of segregation. We also learned why Correns called segregation Mendel's first law. I hope this lecture helped you understand the foundation of classical genetics in a simple and easy way. Thank you for learning with Hello Biology. Keep learning, keep growing, and keep exploring the fascinating world of biology. See you in the next lecture. Don't forget to like, comment, share, subscribe. Thank you and goodbye.